Anyway, so I'm going to focus on stability and resilience, as my title suggests. I'm greatly going to benefit from all that you've already heard, such that I don't have to or shouldn't say much about background. And you'll see that many of the issues have already been raised in a different way, shape, or form, including really most of this, which is the now still incomplete list of the benefits that we think we accrue from our own microbial inhabitants and microbiome, as it's called. You've heard about some of these. Peter gave a wonderful talk earlier this afternoon about xenobiotics and xenobiotic processing. There's been allusion to metabolic regulation and co-metabolism in some of the other talks here. We've talked a little bit about the immune system, and Eric even alluded to the whole business about colonization resistance to pathogens, the whole idea that the microbiome plays this important and still as yet really poorly described and understood role in preventing the invasion of the system by organisms that truly make it their business to cause pathology. So this is now one of the classic studies, and in fact, even though this is 1962, this work really began in the mid-50s, soon after the discovery of antibiotics and the commercialization and commercial production of them. This paper and work like it referred to this whole idea of competitive exclusion, the idea that organisms that share the same ecologic niche might compete for resources, and in this way, a system that is robust in the way in which ecologists think of robustness excludes organisms that would otherwise compete and operate in the same niche. I just want to point out there is a history to shininess as a possible benefit to us. It now goes back at least 20 years. This is a single that came off of the 1991 REM album, Eric. I just wanted to point out to you, they talk about shiny, happy people. And of course, if anyone knows the history of the words to this song, it's actually quite a sad story because it's a play off of a Chinese propaganda poster that, anyway, came out of Tiananmen Square. So the thing about mutualism is that it's a two-way street, and we haven't talked as much about the benefits that microbes accrue from their association not only with each other but with this human host and in fact animal hosts. And there's a lot that could be said. I am probably not in a position to spend any time talking about this, but these are important issues as well. And of course, you've now heard a fair bit about the structure, at least the phylogenetic structure, and in some ways the functional potential composition of the human microbiome. And one of the striking things that is interesting about animal-associated microbiomes is the peculiar nature of the structure, either phylogenetic or even compositional in the way of talking about functional genes, et cetera, that distinguish them from the microbiomes of the external world. And these two kind of tree analogies are ones that I like to think about because they refer to the fact that on the right, the animal-associated habitat microbiomes, there are relatively few deep lineages, and yet a world and wealth of very poorly explored diversity that you might think of as all kind of flourishing at the fine twig and small branch level rather than at the deep branch level. And Eric also referred to that as well. So again, here are the questions that I just want to pose and put out on the table. I think I'm going to have more to say about what might be and less to say about what is. But these are, I think, some of the important questions right now in our study of animal-associated microbiomes. How robust are they? Meaning, how resistant to change when faced with and exposed to various kinds of disturbance? How resilient are they? Meaning, how well do they recover from a disturbance? And how stable are they, even in the absence of an obvious perturbation? And as I will end in discussing very briefly, I think these are also clinically relevant questions, and Julie did a great job in trying to draw in the clinical relevance of some of the questions that we might be asking from a more purely theoretical or sort of basic science-based approach. This fellow, Buzz Hollings, was one of the first to talk about resilience in ecologic systems. He published a series of papers back in the early 1960s. One of them is well worth your read. It's shown here. This is actually a 1973 review. In it, he defines it from his perspective, what is resilience, and he talks about it as the capacity of a system to absorb disturbance and perhaps reorganize, but still retain essentially the same function, structure, identity, etc., that it had previously. Now, this, of course, begs the question, well, what are those functions that we care about, that we're talking about when we try to assess resilience? And I'll, again, return to that as well. I think that's still one of the big unanswered questions. What are those ecosystem services about which we think we might care and want to care because we'd like to know, are they preserved or do they recover, or are they somehow harbored in equivalent kinds of organisms that haven't yet really been fully identified? One of the other interesting things about this paper and Hollings' work was his drawing attention to the distinction between resilience and stability, and he talks in this paper about the trade-off in some systems where rigidity, meaning the appearance of high levels of stability, is actually associated with lesser degrees of resilience. It's almost, you can think of it as a brittle system. It absorbs disturbance to some degree and then breaks. Whereas systems that have a lot of dynamic behavior and appear to be modestly unstable, in fact, have resilience. So Les Dethlefsen set out some years ago to set up some clinical studies to look at the effect of an antibiotic as a disturbing factor on the human microbiome. We picked adults who reported no previous antibiotic use in the prior year. Les, as I sometimes joke, but this is not really all that funny, found about 12 people in the San Francisco Bay Area that hadn't had an antibiotic in the previous year, and enrolled them, and then began to collect fecal samples. And at the time the study began, we were just collecting fecal samples. With the time intervals or timing that you can see from those vertical white lines, there's a 2-month period prior to the first exposure of this antibiotic, ciprofloxacin, then 6 months of observation, and then another exposure identical to the first, and then 2 more months of observation. And when we began to look at these data, there were a couple objectives, one of which was to describe the nature of the disturbance from Cipro. Another was to describe the degree to which there appeared to be a recovery when measured in various kinds of ways, and then to be able to predict these kinds of phenomena by learning what might be the features that are associated with subsequent recovery, etc. And Les used the methods that have already been alluded to or gone over sort of in passing here. This is a taxonomic compositional assessment look. These sets of data here. So we're looking at 16S ribosomal RNA genes and sequencing a fair number of representatives of those genes in these samples over time. And these are data from 3 people. So Les first looked at the first 2 months. Nothing has been done to them, we think, and they're recording some of what we thought would be the important clinical daily features of life, nothing like the iApp, which didn't exist at the time this began. And what we saw, and I'm sorry it doesn't project well, were relatively stable-looking taxonomic compositions over that 2-month time prior to Cipro. But you can see some bumpiness as we come up against that first vertical line, which is the beginning of Cipro. And the bumpiness simply reflects the fact that Les had decided to sample more frequently as we got close to the time of that 5-day first exposure. So that then caused us to think, well, how one assesses and sees stability is a function of how frequently one looks, and of course, as well as how one looks at these samples. And so Eric has actually introduced this whole idea of whether one sees different kinds of phenomena depending at the level of time resolution one looks. So Les has recently gone back to look at some public data of a very similar sort to that which Eric has described. These are data published by Rob Knight's group. Greg Caparoso is a graduate student in his lab. Graduate student or postdoc now? Maybe postdoc. Anyway, last year published in Genome Biology, sets of data from 2 adults that did daily sampling. This is not just fecal sampling, but also tongue swabs and skin swabs. And one of them sampled for 15 months, the other 6 months. And these are the composite data with the first 2 components of a PCA. And they're color-coded according to body habitat from which the sample came. Green are the tongue swabs. They're nicely, tightly clustered on top of each other up there at the upper left. The pink are the skin samples. These are from actually palms, both palms left and right. And then the poop samples are the blue ones down there. And this reproduces the now well-known habitat specificity of the compositional states of the human microbiome, and highlights the fact that it's habitat, gross habitat, that is, if not then one of the most important sources of variation. Okay, now, if you plot the same data, this is straight out of their paper, against time on the x-axis, and the plot on the y is the value that was generated from the first principal component, PC1, you see, because you remember PC1 separated out the samples by habitat, you can see separation here now over time, a long time. And what you see here is that over time, as you saw in the previous slide, there is coherence of habitat and the taxonomic state. Okay, these are the previously published data. Les Lefson looked at these data in a slightly different way, and first did so by calculating an average community composition for the whole 15 or 6 months in these 2 people, and then plotted each time point against that average using a commonly used ecologic distance. It's the Bray-Curtis distance, or log thereof. What you see here are these distances to the average for each of these 2 individuals. The 15-month one is the blue, and the 6-month one is the purple. And the beginning of the year clearly shows something was going on. There's a change in the distance to the average over the whole 15-month or 16-month period, and then it sort of settles down, and you have a relatively similar distance to average for the rest of the 15 or 6 months. So we looked at this for quite some time. Les did. He plotted it with many different kinds of ecologic distances, and did so not only just for the fecal data, which is shown here, but for the tongue and the palms, and saw the same thing, to lesser and more degrees. So he finally called up Greg and said, can you tell me anything about what might have been going on in these 2 people, maybe just before the collection began? And although their IRB has asked them not to reveal this information, subjects can reveal information. And so we know, for instance, that both of these 2 people, before this collection began, went to Asia and, in fact, developed diarrhea and took Cipro, as it turns out. And their diarrhea improved, and they stopped the Cipro and came back home, and they started sampling the day after they returned from Asia. So Les said, maybe that explains what's going on here. Let's just take out the first 45 days or so, and then look at the rest of the data now with a slightly different kind of comparison. This is what some of you will probably know well is a semivariogram, where now we're plotting the same ecologic distance here on the y-axis, but on the x, it's the time interval. It's the duration of time between any 2 pairs of samples, starting with all pairs of samples that are separated by 1 day at the far left, and then all the way out to the right are those pairs of samples that are separated by as much as 300 and something days, or close to 400 days, and it gets noisy because there are fewer and fewer pairs with that kind of time separation. The point here that one might see is that that this plot line, although noisy, is not a stable distance. it's an increasing distance, slowly and inexorably increasing distance the further apart any two samples are. And I'll show you, or one could show you, similar kinds of plots for the tongue and for the skin. The tongue increases at almost the same rate, the palms not so much. But what this suggests is that there is, over time, a form of drift in the degree to which any pair of samples from the gut, the tongue, the palm are similar in their taxonomic composition. So maybe things are not absolutely stable over time. There is a phenomenon going on that reflects some other kind of process. And these are, again, adults. You also might notice that way down at the shortest time interval differences between any pair of samples, there's an even sharper increase over these time interval periods before it settles into that second slope. And this is something down here at the origins that we think might reflect the temporal autocorrelation phenomenon that Les and others have described certainly for fecal samples, maybe for others as well, where sampling very close together in time reflects the lack of true independence of the samples, but rather there's sort of the resampling of a system that's a flow-through, but is sort of an averaging of phenomena that are going on over what might be a 3-day period in the case of normal transit time in the gut. When you do an ordination on the same data, again, the Caparosa data, and now color code the samples based on roughly 45-day periods, shown sequentially moving down in color, you see primary clustering over here on the right and moving down as this blue arrow would suggest, but a bunch of samples that are scattered off towards the left. And if you look at the taxa that are accounting for PC1, you find that it's primarily a Bacteroides-dominated community that's found over here on the right, where this particular person is spending most of his microbiota's time. But occasionally, and this turns out to be a day or two at a time, maybe a handful of times over each 45-day period, there's an excursion out to the left, and that's a Prevotella-dominated community type. So these are actually somewhat similar to two of the meta-hits described enterotypes. And although this doesn't prove anything, it would only suggest that maybe any given individual, if you look closely enough in time, spends a few days on occasion with a different enterotype than they spend the majority of their time as. So it reveals certain subtleties, perhaps, that we think might be real to the stability of the microbiome over time that might not have been seen if you were just measuring weekly or monthly or didn't have that many time points with which to do this kind of analysis. So I just want to again return to this idea of response to disturbance and highlight again these questions about what is it that allows a system to recover if it does recover, and what does that tell us about the nature of a system that might be both predictive and allow for interventions to perhaps restore this kind of feature. I think sometimes useful to think about fitness landscapes and to think maybe clinically as clinicians about where your patient or your own microbiome may sit on one of these adaptive landscapes at any given time, because the topology of that landscape has predictive value in understanding the likelihood that community, the little ball, is going to find itself sort of falling into an alternative state, a different little valley, and the degree of force or disturbance that would be required to shove it into a different valley. These little plots over time are examples of degradation of the landscape by human activity, causing the topology to change and these likelihoods to shift as well. So again, the kinds of things that you may hear about from Larry after me might be sort of conceptualized by thinking about these landscapes and whether we could and how we go about describing these sorts of landscapes for any given individual as a way of understanding what will be the behavior of the system. There are two ways in which we've been looking at disturbance. One is the antibiotic exposure. The other is tooth cleaning, How am I doing for time? Okay, good. So just quickly, this is unpublished work by Katie Shaleff, who's a graduate student in my lab, an ecologist by training. She's looking at mechanical disturbance on the subgingival tooth surface and pocket and asking what is the site-specific behavior of these communities when the dentist does that horrible thing that you've all experienced, where they try to scrape and hack away as much biomass as they possibly can. They think that they are resetting the system as well as reducing biomass, and they would like to think that they are doing so in a way that restores the system to a more pristine, original, ecologic state. Okay, so the question is, are they? And is there anything to be learned by this? So here's the setup. Katie has studied 4 people with healthy mouths and healthy sites, 4 people with moderately severe chronic periodontitis, that's bad gum disease, and 4 sites within their mouths with moderate disease as well. There's a time course at each site. Each site's sampled separately with these paper points, fairly non-destructive, and we have kind of time series data for each of these 4 sites and 8 mouths. So I'll just show you this one thing here. Each of these red dots is a site in one of the periodontitis mouths. Each of the blue dots is a healthy site in one of the healthy mouths. And this is their starting state. This is 14 days prior to the mechanical disturbance. This is now where the compositional state is at 1 week, 1 day, and then the day of cleaning. So that's 2 weeks, normal oral hygiene, a fair bit of variability in the taxonomic composition of the communities at each of these sites. Each site is somewhat distinct, yet those disease sites are behaving and operating in a kind of ordination space that's distinct from the healthy ones. And these are representative mouths of the other 3 for each type. That's all what happened before disturbance. This is the mechanical disturbance, day 0. They're now going flying off out here a little bit more to the right. The red ones do their thing. The blue ones do their thing. By the way, this is now just 1 week after that horrible procedure that you have. There's a red guy down there in the blue space. This is probably the one bit of good news that we have for the dentists, because they would love to think that all these little red sites are being shoved into blue space and staying there. They're not. They all tend to go back to roughly where they were, in some cases by 3 days, in some cases by 7, but usually by a week anyway. And so what we've learned, and Katie's looked at, of course, at all of the sites and all of the mouths, is, whoops, let me just go to the next thing, is basically on average a disturbance that is more distinct and dramatic in the disease sites than it is in the health. It explores a distinct space after the disturbance with the disease sites than it does with the healthy sites. There is return by, on average, say 3 days or so, or maybe a week in each of these 2 systems. And the yellow are just the taxa that are represented on a biplot that account for the dominant sort of sources of variation, or the kinds of variations seen in this particular PC1 versus PC2. And the interesting thing is these disease sites are starting over here, and they're reflective of taxa that have been associated with disease, largely. They go out here and are made up of more organisms that have been associated with health, like Neisseria and Haemophilus and Kingella, but then they go back. And likewise, the healthy things go back to where they started as well. So there's site specificity, disease and health specificity to the nature of the response. The greater the degree of inflammation at the starting point, I didn't show you these plots, the larger the displacement. So we can predict the magnitude of this displacement. They all seem to recover back to where they were. And the bigger question is, if we have more data with more starting states of different types, can we begin to learn what might be the predictive features that say this site actually will now end up in an alternative state, whether it be good or bad? We wouldn't of course know yet. And these are the features that predict that the site will return to where it was. And likewise with the antibiotics, these are published. I'll just go through very quickly. Starting state, 2 months of data that I showed you already for the 3 individuals in this particular paper. 5 days of Cipro. The 7 days after the Cipro has stopped, the taxonomic state is beginning to recover, although to differing degrees. The end of 6 months, D up there in green has fully recovered, E and F not. The next Cipro exposure 6 months later, similar kind of disturbance, partial recovery, especially for D, which now is nowhere near as close back as it was before. And now the end of the study where D has not recovered as opposed to its first exposure. So they're all different taxonomically, but they responded differently depending on whether it was the first or second exposure. We also looked at metagenomic data on actually now 3 subjects, a smaller number of the samples, trying to learn what are the functional genetic features that are associated with either recovery or not. And I'll just tell you that in pink here, there actually is purple probably up here, is the this is another ecologic distance shown for the functional compositional data versus the taxonomic compositional data in light blue. We're using the same data sets for both of these, and you can see the disturbance is greater taxonomically than it is functionally. There is largely recovery both ways, but the recovery is more complete with respect to the functional state. And we're still analyzing data and trying to get to more people so that we have a larger N of individuals from which to learn what might be the functional features that are associated with disturbance and recovery. But we know from the classic ecologic literature that there are a number of features that have been classically associated with recovery, including redundancy, including degrees of richness, the nature of the evenness in the patterns of organisms and their genes, the nature of the interactions, and then the timing of how they respond, at least in well-defined, somewhat simple systems. So this is clearly way early to be able to say we understand how these systems are responding and what are the predictors of response. But again, what I think would be great would be able to say, we've analyzed a bunch of sites, we've done a profile of a mouth, we've looked at the gut in a person who's about to undergo a known disturbance, and we can predict what the outcome will be and how we can manage the potential adverse consequences better. So I will leave with these conclusions here. Basically, really distinct, interesting ecosystem that is the human. There is relative stability, but you saw it dynamic and drift behavior over time. There is relative recovery and resilience under the conditions that I've described in the natural state. Clearly, history matters. Subject D was an example. And, you know, again, what are the services that we care about for which we're saying there either is or is not recovery, or for which we think there, you know, we need to see recovery in order to have host health? Those things are not yet well described, and that's what I think we need to be working on, among some of the others. So I'll just mention Lefrufson again on the Cipro study, Katie Sharef on the mouthwork I showed you. Gary Armitage and Peter Loomer are wonderful dental colleagues at UC San Francisco, without whom none of this would be possible. Susan Holmes is a wonderful statistician. Some of you may know her. We've been working with Jill on work that I had hoped I would be able to show and haven't, but maybe we can talk about in the discussion period later, looking at the assembly of organisms in the gut and the importance of strain distinctions and strain resolution. And then the rest of my funding agencies and your attention. So thanks.